All right, I want to talk to you a little more about PCR. So, we're going to go through applications of PCR. Excuse me. So, different things. We have DNA microarrays, or what they call DNA chips, right? So, DNA microarrays or DNA chips. So, essentially, what they do here is we use PCR to create probes. So a probe is like a primer. So a probe is a small complementary piece of DNA. So it's much like a primer. So really a probe is used to what we call fish out DNA. So if it's complementary, it'll anneal, right? And then we take that DNA, and we take that probe, and we attach a fluorescent marker. Right, and we attach a fluorescent marker. So these were once thought to be the future of pharmacy. There was a emerging field called pharmacogenetics. Where we thought we could personalize medications based on DNA. Originally, it didn't go very far, but recently I had a family member who was taking medication, and the medication wasn't seeming to work. And they did a DNA microarray study, and they looked at DNA markers and said, hey, this medication probably works really well based on his marker, and this medication probably doesn't work so well because of the DNA markers. So they use fluorescent probes. And what happens is if you have a complementary piece of DNA, it'll light up a marker, and that marker will tell you that maybe you have a proto-oncogene or a gene that could become cancerous, or if you have an oncogene, a gene that's already cancerous. So you could do it like that, or you could do it for medications, but essentially all this complementary DNA that is produced by PCR is attached to a plate, right? And then we can simply wash DNA samples over it, and if it's complementary, it'll light up. So DNA sequencing is a big use, right? So DNA sequencing takes advantage of DNTPs or DDNTPs, right? And the DDNTP stands for dideoxy. So remember, if we have a ribose, right? So the normal DNA is deoxy at 2. Right? Deoxy at 2 prime. The dideoxy then is deoxy at both, right? And remember that hydroxyl right here, right? And this is your 2 prime and your 3 prime. Right, this was your 1 prime, 4 prime, and then we have a CH2 up here that was 5 prime. And this was your 5 prime. Right? So remember, it's made 3 to 5, but if it's deoxy here, I can't make it. So it's called the dideoxy chain termination method. Right. So attached to each dideoxy is a fluorescent marker. Right. And we would have done spectrophotometry by now a bunch, and we use spectrophotometers to read the fluorescent markers. It will give us the sequence based on the color, right? So each dideoxy has a fluorescent probe, right, attached to it, right? And then each, like A is green, red is T, blue is C, right? So and black is G. So these spikes on these mass spectrophotometers will go ahead and give us the sequencing, and that's done by machines, right? So you don't have to do that by hand. Spectrophotometer will read those first. So dideoxy. So we have a mix of both DNTPs and DDNTPs. If it gets a DNTP, it continues the chain, and every time it reaches a dideoxy, it terminates it. Okay. And I have better pictures than what I drew right up there. Right. We can also use in medications or for diagnostics, PCR in medicine. And we already talked about these. So we can use it for bacterial infections. We can do DNA barcoding, right? So both of these take advantage of qPCR, remember, or real-time PCR. Bacterial infections, we do what's called DNA barcoding, and we look at the DNA. We look at the DNA in the small ribosomal subunit. So they're all slightly different, right? But we can tell the different bacteria, and the reason why that's important is certain antibiotic-resistant bacteria will have a different DNA profile than ones that are susceptible to antibiotics. And then viral infections, we can use RT-PCR. Zika, right? There's commercial tests available for Zika. And then we know that COVID, right, is performed by RT-PCR followed by the qPCR again, right? So we already talked about that the other day. We can use it for testing in GMOs, right? Remember, for GMOs, we used our multiplex. We had a fantastic GMO lab we would have done by now doing PCR, but we're not in school right now, so we didn't get to do it. So there are a number of different, we have BT, Bacillus thuringiensis, we have TI, which is A, tumor fascians, common bacteria that are able to insert their plasmids into plants. So we use those to insert genes of interest, right? So if you look at here, so these bacteria are common to plant communities. So when I do a PCR, I want to look for the promoter, the terminator, and the gene, right? And the reason why I want to look for all three is I might have a promoter and terminator that's common to that bacteria that would infect plants. So just because a plant has the promoter and terminator doesn't mean they're GMO. But if they have the promoter, the terminator, and the gene that we're looking for, then they are GMO. So we have suspected GMO and confirmed GMO. So a probe, we make a probe, which remember is the same as a primer. We'll use conventional PCR. Right, real time will work. And then we can do this followed by gel electrophoresis. So if we don't have real-time PCR, I can do conventional followed by gel electrophoresis. So remember that using multiplex PCR. We can look for transgenic animals. Remember, farming is creating transgenic animals. I don't believe that we're currently doing that in the U.S., so not really good application. Probably one of the largest applications on the commercial basis is PCR forensics. So a couple things. We can use it for AVO typing. There are probably simpler ways of doing it, so it's probably not that common. But DNA fingerprinting, originally called southern blotting, because it was developed by a guy named Edward Southern, took advantage of what's called RFLPs. And they were repeat fragment polymorphism, so long strands of repeat segments. And the patterns for those are usually unique within families. There's a problem with using RFLPs, and the problem with using RFLPs is they're very long, so they don't PCR very well. And they're not all that specific. They're pretty specific, and specific means unique to an individual, but not completely. So the discovery of things called STRs, right, short tandem repeats. Now, short tandem repeats are less specific than RFLPs, but I can look at more of them, right? So I can use my STR analysis to make it highly specific and fast if I use a number of them. Okay, so RFLP is about 1 in 3 billion. Now, 1 in 3 billion is probably pretty specific, but if you think we have over almost 8 billion people in the world, it's probably not specific enough, especially if you're convicted of something you don't think you did. So we generally use STRs, and both of these are NTRs, or non-translated regions. So STRs by PCR. So short tandem repeats are 2 to 6 base pair regions within your genome, and there's a lot of them, right? But currently, the law enforcement looks at 13 loci. A loci is a position, right? So currently they look at 13 loci. And if you look at this simple picture, so these short tandem repeats, not everybody has them, but everybody has different amounts, right? And the pattern of those different amounts make what is unique. And they're on the different chromosomes, right? So they're inherited individually. There's a lot of variation. So if I look at 3 loci, the chance that 2 individuals will be the same are 1 in 262,000. So it's not all that specific. But by the time you get to 13 loci, right, it's 1 in 575 trillion is the chance of having 2 people identical. So it is really specific. It can be really fast. It's something you could use multiplex for. But the CODIS, make sure you remember the name because I think it shows up. The CODIS is a storage system used by the FBI and national law enforcement agencies. And whenever you're convicted of a felony, your DNA is swabbed and it's put into the CODIS. So if your DNA ever shows up at a crime scene and you're in the CODIS, then you're automatically a suspect. Now the problem with that obviously is you leave DNA everywhere, and just because your DNA is found at a crime scene doesn't necessarily mean that you were the one who did it. It just might have been unfortunate, or you could have done it. All right, so other uses of PCR, we can use it for paternity testing. I lost my pen. And that would use STR as well. Originally, it was RFLP analysis, but now it's shifted to STR. So if you look at this picture here, you got mom and dad. These are looking at STR regions. This one is definitely, daughter 1 is definitely a child. Daughter 2, mom's got some explaining to do because dad's pattern of STRs do not match. Son number 3, this is a yes, this is a no. This is yes. And this is adopted. Right? So you can look at patterns to find for paternity, either adopted or switched to the hospital. Another uses of PCR, you can do mitochondrial analysis. Remember, mitochondria are only inherited from the mother, and mitochondria are endosymbionts, so they were evolutionary captured bacteria. So, since they're only inherited from the mother, we can use them to show a migration study. So when they say that human population started in Africa, then they're tracing these mitochondrial lineages back to Africa, right, for common ancestry. We could also use STRs for animals, right? So we can look at illegal remains when poachers are arrested to find out where the species came from. So that's basically it, and I will talk to you soon.